Sure. Muscle milk helps build muscle. One that matters most. The muscle. And almost had it right. They just don't have it in the right order. And at number 5, I'm going with the Buffalo Bills. The last 2 weeks, Bills fans have been on me like they're the Rodney Dangerfield of my power rankings. And they think that I don't give them any respect. And so now I feel like I got to show you how to do it the right way, Buffalo Bills. The way that you played against the San Francisco 49ers, that's the way to do it. This makes me put you in my power rankings. This makes me not give you the disrespect of a Black Thought, of a Beanie Siegel, of a Master Ace. When you have Cole Beasley, when you have Stephon Diggs continuing to get open, continuing to make plays, Tre'Davious White now coming along, 3 interceptions in the last 4 games, Edmonds at the linebacker. This is the team that you don't want to play in the playoffs. And as they head down the stretch of the AFC East, they have an opportunity to make a ton of noise in the playoffs with the way that they play football and a quarterback that's efficient in the red zone and tough to stop inside and outside the pocket. And number 4, I'm going with the Green Bay Packers. Why? Aaron Jones, Aaron Rodgers. The only thing that can make this offense better is if they had Aaron Neville singing before every game. Yeah, shout out to the NO. Aaron Rodgers is playing absolutely out of his mind. He's playing at an MVP level, a level we haven't seen since 2016, 2014, when we thought he was the best we'd ever seen play this game. And look at what the things that Aaron Rodgers and Aaron Jones can do. 77-yard touchdown last week to seal the game, catching the ball out of the backfield. Obviously, we know who Devontae Adams is and what he can do. And at number 3, I'm going with the Pittsburgh Steelers. And yeah, they're going to be a little bit mad this week because I had them at number 2 in the previous weeks, and they were undefeated. And they were like, "Oh, Ryan, you lose your Steeler flag. You don't get to raise a terrible towel." Shut the hell up. I knew this jump was coming. I saw it coming. This team, they didn't need a loss, like Bart said, but they were on their way to losing. It wasn't executing offensively. They couldn't move the ball in the run game. But Ben Roethlisberger has been amazing this year. He's emerged. If it wasn't for Alex Smith, he'd be the comeback player of the year. And so when you watch this team and you see this defense losing some pieces.
Hey, Kayla.
Hello, can you hear me?
I can hear you, yes.
Awesome.
You in the weight room?
I'm in the HES lab.
Oh, okay.
Yeah.
Hey, Ben.
Beth says hello, Dr. B.
Hey, Beth.
He says hi.
Hey, Ben.
I've been doing good.
I've been getting your quizzes that you've been making up, so thank you for making those up.
Yeah, I feel bad that it took me a really long time to get them done, but I'm finished with them all now, so I just have the exam to focus on.
I think what I'm going to do for the exam, getting all that loaded on the Blackboard is going to take hours.
Yeah.
Furthermore, when I started typing some pieces that Blackboard doesn't like. So when you put it in and then you save it, it changes them.
Oh.
So I might just make it a paper test and then email it to everybody and then say, you have the 2 hours, finish it, email it back, and then just do it that way. Because the Blackboard thing is looking like it's not really going to work. So.
Okay. Gotcha. Oh, yeah. I didn't know that you were planning on doing it on Blackboard. I just thought it was going to be a paper test like the other ones too.
I think that's what I'm going to do. I've got so much grading to do, and to spend that much time on something that may not work.
Yeah.
No, thanks. No, thanks. So, did you get a chance to look at the reproductive system?
I have not done that part yet. I did watch the video for lab, so I saw like that whole hour-long video. But other than that, I haven't gotten to that lecture yet.
Then yesterday I did the bit on pregnancy and delivery.
Okay.
Reproduction, the hard part's the endocrinology.
Okay. Yeah.
I'm perfectly confident that you and Emily know about the structure of ovaries and the uterus and what have you. I don't think that's a problem for you guys. It's more or less the menstrual cycle, the regulation, and then the changes that occur during pregnancy.
Okay. Sounds good. So
Do
make
you have
sure I
any
look
questions
over.
about, let's back up. Do you have any questions about the urinary tract, the urinary system?
I don't think so. I haven't. Yeah, I have to catch up on some lectures because I had a couple PT interviews last week and some projects to finish up. So I was having this whole week to focus
Hey,
on
how'd those go?
really well. I actually got accepted to Trine University, so that's exciting. And
Congratulations.
then, thank you. I had an interview with Mayo Clinic last week too, so I thought that went pretty well, and just waiting to hear back from them. So,
Where's Trine on your list of desired schools?
it's definitely up there. I like the program, and it's in my hometown, which would be nice. But it's definitely actually, it's probably the most expensive school that I'm looking at. So if I could get in somewhere where like even living somewhere else would be cheaper potentially, I would probably pick a cheaper school over them. So yeah.
Interesting.
Yeah. I mean, the program is good, but I've definitely had a lot of PTs be like, it doesn't really matter where you go to school because they all have to teach you the same things, and like getting clinical hours really matters, which Mayo Clinic does more clinical hours than other places. So that would be a selling point for them.
And you're all
But yeah,
set for a national exam too.
yeah. So
Remind me, does the PT exam have a practicum as well?
I'm not sure. I don't know if the boards have a section where you have to do the practical part or not. I haven't looked into that yet.
Because in medical school, you do the national boards,
Mhm.
and for the practicum, you have to make an appointment. Then you have to fly there
Oh yeah.
at your expense.
Oh wow.
And now with shutdowns and what have you, a lot of that's being put off.
Oh yeah.
So these places need doctors, and the ones who've been trained can't get their practicums to get licensed
Okay.
because of the shutdowns. It's kind of a
Gotcha.
mess.
Oh yeah, that makes sense. Yeah, I don't think there is anything like that for PT then. I haven't had any physical
Is
therapists
that
tell me like about
is that
that.
Mitch I hear in the background?
No, it's Jake.
It's
He's
Jake,
pretty
okay.
good back.
Because if Mitch was there, I said you could just ask Mitch. But
Oh yeah, that's true.
all
But
right.
yeah, I
So
don't.
the kidney, it's very important that you're able to break down the nephrons into the different sections: proximal convoluted tubule, descending loop of Henle, ascending loop of Henle, DCT, collecting tubule, and the DCT is divided into the early and late DCT. The late DCT is a lot like the collecting tubule.
Okay.
So
Can you guys see me?
physiologically, in fact, those two are kind of sort of the same, physiologically speaking. They respond to the same hormones. They have what happened to Emily?
I think she's trying to get her video to work.
Okay. They respond to the same hormones. They have the same ion pumps and what have you. So,
The DCT and the collecting
yeah, the
too.
collecting tubules. So they are very much alike. You can almost think of them as extensions of each other, physiologically speaking. It would be fair to think of them as extensions of each other. PCT does probably most of the heavy lifting, and you recapture things by a combination of secondary and primary active transport.
Okay.
Secondary active transport predominantly at the apical surface, primary active transport predominantly at the basolateral surface.
Okay.
Then the loop of Henle, the descending limb is permeable to water, impermeable to salt. The ascending limb is impermeable to water, permeable to salt.
Okay.
And then the thick part of the ascending loop actively transports salt from the filtrate into the interstitial space. And it uses that very powerful NKCC2 pump.
Okay.
Emily, can you hear me, or is your
I
video?
can hear you guys. I don't know why my camera is not working. I don't know.
Go down to where it says video. Click on that, and it will give you a choice of cameras.
Yeah, the only option is my FaceTime camera, which is what I always use, and it works, and now it's not. I don't know why.
Because my camera just didn't work once, and I unselected it and then reselected it, and it started working.
Okay.
I don't know why, but
I'll try. I'll go to video settings and see.
or just shut the video off, turn it back on, and see if the camera comes back on.
Yeah, that's what I've been doing.
And still no luck. Well, we can hear you. I mean, it'd be nice to see you. But
I don't know. So weird.
computers. Can't live with them, can't live without them.
I don't know.
All right, so then the DCT, the late DCT is still impermeable to water, and you have more specialty ion pumps there. And I talk about those in the lecture. And then in the late DCT, the late, again, is a lot like the collecting tubule. You have alpha intercalated cells and principal cells, and their ion moving capacities are regulated by aldosterone, first of all, and then the presence of aquaporins is dictated by ADH, which influences how much water gets recaptured from the filtrate. So those are the things on your checklist to review.
Okay.
And the last bit, which I really didn't get to talk about very much, is urea. So urea is the main non-ionic solute that the kidney handles. So amino acids, when they are deaminated, that produces ammonia. Ammonia is quite toxic to cells. So the cells with the liver mainly, but other tissues do too, but the liver is the champion of this, converts ammonium into urea. Urea is very non-toxic. So urea is filtered, but then urea gets pumped into the interstitial space by the loop of Henle. Then it gets secreted back into the filtrate, then it gets pumped back out. So it gets recycled actually. So it gets pumped back in, pumped back out, pump back in, pump back out, pump back in, pump back out. That's called urea recycling. Okay.
So the loop of Henle pumps it into the interstitial fluid, and then what pumps it back in to the loop of Henle?
The ascending loop of Henle pumps it back in, and then the collecting tubule pumps it back out.
Okay.
So it goes into the interstitial fluid via the collecting tubule. There's a transporter called URT1, the urea transporter 1, and that keeps pumping it in. It just keeps getting recycled.
Okay.
Some of it doesn't get recycled. Urea is the major nitrogenous waste in urine, but that recycling, that's how you regulate solute concentration in the medulla. So you have mainly sodium and urea and chloride as your main solutes. That gives you the gradient, solute gradient in the medulla. It's mainly sodium, chloride, and urea.
Okay.
So go ahead and look over that.
Okay.
Emily and I went over that in pretty gory detail on the Q&A I did with her.
Okay.
So Emily's the expert. If you get lost, just ask her.
Okay.
I can show you my pictures that I drew.
Okay. Yeah.
Yeah, I
that'd
forced her to draw some pictures,
Yeah.
and she's actually a better artist than I thought she was.
Oh,
be-
thank you.
So they're on her iPad, so I think she kept them.
Yeah, I can send them to you.
yeah,
Yeah.
that would be great.
And with processes like this, if you're a very visual learner, and Emily confessed to me she's a very visual learner, I told her, well, then Emily, draw pictures. So I had her draw me a picture, and that I think really
Yeah.
concreted it in her head. So if you're a visual learner, I'd ask you to do the same.
Hmm.
That definitely helps to draw pictures.
Yeah. Do you recommend any like video sites like Khan Academy or anything for like
Khan
also
Academy,
quick
the
description?
problem I have with Khan Academy videos is that they're mealy mouthed. They go, for example, the one they have on the kidney, they'll talk about the PCT, and then they'll go back and re-talk about glomerular filtration.
Hmm.
It's like, dude, you just talked about that. So they take 20 minutes worth of material and they stretch it into 60. And I think students get so bored, they turn off after the first 10 minutes because they spend so much time repeating themselves.
Hmm.
That's my problem with Khan Academy, is they get mealy mouthed and they lose people.
Okay.
The other piece about Khan Academy, and I didn't know this until I talked to a friend of mine who actually did some work for them.
Mhm.
They just talk. The drawing is actually done by a professional artist.
Oh, okay.
So they work with a professional artist who draws while they talk.
Hmm.
So it's not, my first thing was, David, I didn't know you could draw like that. He laughed out loud. He said, that's not me.
Hmm.
They hire professional artists who do the drawings, and then they sit there with you to make sure that they have you try and draw it, and then the artist cleans it up,
Hmm.
and then they draw it, and then they do it while you talk. And he said, you have to do it several times because the artist is not a scientist. The
Yeah.
artist is just trying to draw what you say.
Mhm.
So it's a professional artist who does that. And sometimes the drawer gets way behind the artist. So they'll actually speed up the art. That's why sometimes the art will go really fast.
Hmm.
So it's a little deceiving.
Okay.
And that's why you don't have coordination between the artist and the lecturer sometimes. So
Mhm.
I like Khan Academy, but students who've listened to Khan Academy have said they got put to sleep just because it's so mealy mouthed. Their stuff
Okay.
is accurate. It's up to date.
Yeah.
So that's good. Just don't listen to it unless you're really patient. If you're impatient, you want them to get to the bottom line, it'll take a while sometimes. Another good source. Osmosis.org is the best source, but that's by subscription, although
Mhm.
you can get a trial
Okay.
for 24 hours, I think.
Okay.
So you can literally do all the kidney stuff, watch it, and you can watch it, rewatch it.
Hmm.
Their stuff is really good.
Okay. So I was gonna say videos a lot of times like help me like cement processes and see it
Yeah,
happening.
to watch someone else draw it.
Hmm.
With osmosis, the person narrating is usually the person who drew it.
Okay.
So they have a script that was written for them by a doctor or a
Mhm.
physiologist, and then the artist is also the narrator,
Okay.
which is pretty helpful.
Hmm.
I
Okay,
just emailed you those pictures, Kayla.
Okay. Awesome. Yeah, I think I got them.
all right, so that's the urinary system.
Okay.
Um,
And this is the exam's gonna be over like chapters 14 to like 18, right?
the 20, but we're leaving out 15, we're leaving out 19.
Okay.
It's 5 chapters.
Okay.
So it's 14, 16, 17, 18, 20.
Okay. Awesome.
All right, reproduction. I'm sure you guys know all about ovaries and the uterus and blah, blah, blah. Do you have any anatomical questions? You guys are probably fine on structure. So let's turn, go ahead.
Sorry, I was just going to ask, are like main questions on the test going to be about like the menstrual cycle and the endocrinology like behind that?
Pretty much, yeah.
Okay.
All right, so the male reproductive system we did first. So the spermatogonium is the stem cell population at the periphery of the seminiferous tubules. Those cells are activated by follicle stimulating hormone. They're ensconced inside Sertoli cells. The Sertoli cell acts like a nurse cell. They undergo mitosis. One cell becomes a primary spermatocyte, the other cell stays a spermatogonium. The cell that becomes a primary spermatocyte is displaced towards the lumen. The cell that remains basal is always the cell that remains a spermatogonium. The so-called type A1 spermatogony, primary spermatocyte, can divide mitotically. It usually can only divide once, twice at the most. Then it enters meiosis. So it enters meiosis 1, becomes a so-called B2 primary spermatocyte, divides once, enters meiosis 2, becomes a secondary spermatocyte. They stay joined by cytoplasmic bridges. And then they start to differentiate. They become spermatids. Then they go through the process of spermiogenesis. And I talk about that on the lecture, the Manchet stage, that sort of thing. Once they get released from the Sertoli cell, and the release from the Sertoli cell is testosterone dependent, the sperm is still not motile. The seminiferous tubule is lined by smooth muscle. It contracts. It propels the spermatozoa toward the retestes and toward the mediastinum of the testes to the head of the epididymis. In the epididymis, they're stored. And in the epididymis, they acquire motility. It's not until then they become motile. Then the epididymis is drained by the vas deferens. And the vas deferens is also contractile. There's a picture in the slides of the vas deferens, and it's mostly smooth muscle. The sperm go from the vas deferens through the ejaculatory duct. They receive components from the seminal vesicle. Seminal vesicle components are mainly fructose, which is a major carbon source for the sperm. They then pass into the prostatic urethra. They pick up secretions from the prostate, which are mainly citrate. They then pass into the penile urethra. They pick up secretions from the bulbourethral, otherwise known as Cowper's glands. Those are mainly coagulation proteins that clump the sperm together. And then they exit the penile urethra to the outer world. Because of the citrate, and that's what, when we say semen refers to the spermatozoa plus the components of those accessory male sex glands. Seminal vesicle, prostate, Cowper's gland. And because of the citrate, semen is alkaline. That will buffer the pH in the birth canal, which is acidic. Also, the clumping of the sperm protects them, so that you get a core of spermatozoa that survive, even though the ones on the periphery may die. Then the warm temperatures inside the woman's body activate fibrolysins. They degrade the coagulation proteins. The sperm break up and they start to move. And they move towards the cervix. And if the cervix is ready, at that time of month, they can pass. If not, they kind of go boink, 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 and they can't penetrate, and they give up the ghost and die trying, so to speak. That's the male reproductive system in a nutshell. And remember, the urethra passes through the prostate. That's why in older man, the prostate enlarges, and that can constrict the urethra. That's why men, when they get older, sometimes will have trouble tinkling. That's called BPH, benign prostatic hyperplasia, which is very treatable with certain drugs. Women, of course, have a shorter urethra and they don't have a prostate. So women are more prone to so-called stress incontinence and that sort of thing, which you can help with these Kegel exercises. In fact, Kayla, that's something you'll learn about in PT school.
Yeah.
Kegel exercises. Very important. They've been shown to help with stress incontinence.
Yeah, I know one of the PTs at the clinic I work in now is like specifically a women's health PT, and so she deals a lot with like incontinence.
Yeah, they're
and stuff like
not
that.
hard to do. You could do them while you're sitting at your computer,
Mm-hmm.
and if you do them consistently, they do improve with the strength of the pelvic floor. They can help in men as well. They're not as important in men, but men can do them too. And that's an important part, I think, of physical therapy, is that that's an easy fix, so to speak, without surgery or more drastic measures.
Mm-hmm. Okay.
Okay, Emily, how you doing? Any questions?
I felt pretty good with the male system.
Yeah, so the male system, outside the seminiferous tubule, you have an interstitial tissue that contains the Leydig cells. The Leydig cells make testosterone. The Leydig cells respond to luteinizing hormone. The spermatogonia and the Sertoli cells respond to follicle stimulating hormone. Testosterone feedback inhibits the production of luteinizing hormone. Sertoli cells make a peptide called inhibin. Inhibin feedback inhibits production of follicle stimulating hormone. So that keeps those levels in men at a relatively constant level.
Okay.
If you do something stupid like take anabolic steroids, that will shut off your endogenous testosterone, and literally your testicles will shrink down to the size of a peanut.
Oh.
I don't know if you guys are into baseball at all, but there was a baseball player named Rafael Palmeiro, and he took steroids, and he actually wrote about it. He said his testes shrunk to the size of grapes, and he said it scared the daylights out of him.
Yeah, I
But
bet.
it should have.
Mhm.
Yeah.
It should have. He did something stupid.
Yeah.
Yeah. Huh. Is that like the body's response to trying to keep those hormones like level? So if you're taking
Yeah.
outside hormones, it
Yeah.
won't
So
produce any.
you decrease exogenous, you decrease endogenous testosterone production by the Leydig cells, because now the hormones you're taking by mouth, they shut down luteinizing hormone, but because that hormone doesn't cross the blood testes barrier, they're not that without endogenous testosterone, that tissue atrophies
Gotcha.
and your sperm count goes down,
Mhm.
and you become you literally castrate yourself, so to speak. Um, it's kind of scary.
Yeah.
So as far as I'm concerned, he's a bloody fool. But
Mhm.
if you guys see as PTs so-called whole muscle tears,
Mhm.
whole muscle tears before the juicing era were very rare. Now with juicing, whole muscle tears are common.
Yeah.
You see a whole muscle tear in an athlete,
Mhm.
that's usually a sign that simply because the muscle gets too big for the bone.
Mhm.
Yeah.
And that's a sign if you're working with high school athletes, you're trying to see whole muscle tears, you need to say, okay, you're going to go to the clinic, you're going to get a test, and here's what I'm going to tell them what to test for. No, I'm not going to tell you, but you're going to the clinic for a blood test.
Mhm.
And if their testosterone and epitestosterone levels are skewed, that's
Mhm.
how you tell. Because the body turns endogenous testosterone into epitestosterone, your ratio should be around 1. If it's an athlete or a male in puberty or a male before they get older, ratios of, I think 5 is a cutoff. If it's above 5, you're juicing.
Yeah.
It's just that simple. You're juicing. So that's a sign you need a blood test, and that's will be something you'll need to order, because that'll be a sign to you something's wrong. All right, so enough on that unpleasant subject. Females,
Mhm.
more complicated. So let's start first with the menstrual cycle. So remember from the lectures, you probably heard before puberty, the production of gonadotropin releasing hormone is at a constant low level. At puberty, now you get a pulsatile production of gonadotropin releasing hormone. So it now troughs, peaks, troughs, peaks, troughs, peaks, troughs, peaks, instead of this kind of constant, excuse the expression, background noise. So the follicles in the ovary, which consist of a primary oocyte surrounded by a layer of follicle cells that are undifferentiated. It's just a single layer of squamous epithelia. Those are your granulosa cells. There's a picture in the slides. When you look at it, there's actually a layer outside that layer of granulosa cells. Those are thecal cells, THECAL. And those cells, when they start getting increased levels of gonadotropins, in particular follicle stimulating hormone, they start to differentiate and divide. So the first change in the granulosa cells, they go from squamous to columnar. They have junctions that they form with the oocyte. They pump nutrients, RNAs, proteins, electrolytes, metabolites into the oocyte, and the oocyte gets bigger. And then in response to FSH, they start to grow and make layers of granulosa cells. the thecal cells respond to luteinizing hormone. They divide, they take cholesterol, they convert it into pregnenolone via an enzyme called cholesterol desmolase. And then pregnenolone can go one of two ways. You can use pregnenolone to make progesterone, but at this stage, the pregnenolone is used to make androstenedione, which is an androgen. So the thecal cells make androgens. And then that androgen diffuses to the granulosa cells, and the granulosa cells, in response to FSH, upregulate an enzyme called aromatase. Aromatase converts the androstenedione into 17-beta estradiol, which is an estrogen. In fact, estradiol is the most biologically active estrogen. And estrogen is responsible for all the secondary sex characteristics you see in females. So do you guys know what the Tanner stages are of maturity?
I don't think so.
No. So when you go to the doctor as a child, and the doctor has to look at your soft bits, and it was always really irritating. What they're looking at is growth development of pubic hair. They're looking at the growth of breasts. They're looking at the growth in males, looking at the growth of testes and the penis and that sort of thing. And the increase in size is called the Tanner stages.
Oh.
So they're looking for progression through the Tanner stages.
Yeah.
So it wasn't until later that I discovered why my doctor always did that. It always used to irritate me. But now I think I understand what my doctor was up to. And sometimes for PTs, one of the protocols you'll have is if a kid gets a lot of injuries, something you'll be asked to do is to make sure they're at the right Tanner stage for their age. So, and I know, correct me if I'm wrong, some PTs now are starting to specialize in pediatric, or at least adolescent injuries.
Yeah, there's definitely pediatric PTs.
Yeah, so they're having to pay more attention to Tanner stages and Tanner staging and that sort of thing, because pliability, something estrogen does is it increases pliability of, so does progesterone. It increases the elasticity of ligaments. So that's why during certain times in their periods, women are more prone to injuries. And I am sure the trainers have seen that at SAU.
Yeah.
That is well documented, not just clinically, but also with research. But I'm sure if you ask Chris or Brian, I'm sure they'll tell you female athletes get a lot more injuries during that period of their life, what have you. Anyway, so those will be some of the unpleasant conversations you'll have to have with adolescent female athletes. The follicle develops, the granulosa cells make lots and lots of estrogen, and the estrogen will feedback inhibit production of follicle stimulating hormone. And the luteinizing hormone levels will start to then creep up above the levels of follicle stimulating hormone, what that also does, it means that the follicle that's the biggest and most developed, that has the most follicle stimulating hormone receptors, will be the one that survives the others, because they need follicle stimulating hormone to continue to divide. They'll start to regress. So you get selection of just one follicle. Eventually, the granulosa cells will make so much estrogen. Now they go from negatively feeding back on the pituitary and the hypothalamus to positively feeding back on the pituitary and the thalamus. And now you get a surge of luteinizing hormone and follicle stimulating hormone, particularly luteinizing hormone, and that surge of LH causes ovulation. When you eject the ovum from the follicle cells now become the corpus luteum. And that period of life in the ovary, from menses, first day of menses, to ovulation, is called the follicular phase. The follicular phase, which is characterized by a gradual increase in gonadotropins to a spike in gonadotropins, and then a drop-off in the production of gonadotropins. That's the follicular phase. It's about 14 days-ish. Remember, the menstrual cycle varies. Emily, question?
No, I think I'm good.
Okay, your light went on like you were talking.
Oh, it's probably there's background noise in the gym.
Oh, okay. Okay, that's fine. Let's see what else. Then the follicle, the ovary goes into the luteal phase. Now, I didn't mention this. When the granulosa cells get to that point where they're making lots of estrogen, they start to synthesize luteinizing hormone receptors as well. So they respond not just to follicle stimulating hormone, they also respond to luteinizing hormone. That's what causes the large surge in estrogen in a mature follicle. And the follicle also, in response to high estrogen, the oocyte, which is blocked in prophase of meiosis 1, completes meiosis 1 and now gets arrested in metaphase of meiosis 2. And when it's ovulated, it's now arrested. The ovum is arrested in metaphase of meiosis 2. It does not complete meiosis 2 unless it's fertilized by a sperm. Now, those cells of the corpus luteum, they take cholesterol, they make pregnenolone. Now, instead of making androstenedione, they'll make progesterone. And the pregnenolone that is not diverted to progesterone is converted into androstenedione. But then the two enzymes, the 17 alpha 17 alpha hydroxylase and the aromatase, they convert androstenedione to testosterone and then 17 beta estradiol. But they make more progesterone than they do estrogen. Now, the combination of progesterone and estrogen feedback inhibits the production of gonadotropins. So your gonadotropin levels tank during this time. That's a strategy behind birth control pills. That's why birth control pills will prevent your production of gonadotropins, and they'll cause a false luteal stage. They essentially keep the, they fool the pituitary and the hypothalamus into thinking you're in the luteal phase with a combination of progesterone and low dose estrogen. And there's monophasic, biphasic, triphasic, quadriphasic pills, and yeah, blah, blah, blah. Okay, luteal phase. During the production of progesterone, so during the follicular phase, the uterus undergoes a proliferative phase because estrogen is a mitogen for the endometrium. And so the stratum functionale gets thicker and the blood vessels start to enlarge. Progesterone now will cause those blood vessels to become spiral and to enlarge. So they become the spiral arteries. And then the glands, the uterine glands, they get deep and thick and highly differentiated under the influence of progesterone. And they make a lot of mucus that covers the epithelium and prepares the endometrium to receive an embryo. You have to have continuing progesterone to maintain this. This is called the secretory phase. Also, estrogen induces the progesterone receptor in endometrial cells. So it's a classic permissive effect. So without estrogen, you won't be able to respond to progesterone. Then the corpus luteum will last about 8 to 10 days. If it doesn't get any human chorionic gonadotropin, by the 10th day, it will regress. And it regresses and you don't get any more progesterone, and now the secretory phase will undergo the menstrual phase, and that essentially, that appearance of blood is the mark of the first day of the next menstrual cycle. And the average menstrual cycle is 28 days, but it can vary from 20 to 35 days or longer, depending upon a whole host of things. That's the menstrual cycle kind of in a nutshell. So the difficult part about this is understanding that low levels of estrogen cause a negative feedback cycle. High levels of estrogen cause a positive feedback cycle. There's a switch depending upon the high levels of estrogen. And it's about 200 picograms. That's about when you start getting the positive feedback, is about 200 picograms per deciliter is the number that's usually around. Okay, let me pause right there. The slides, Kayla, when you go over them, there'll be lots of graphs that show you estrogen levels, progesterone levels. That should help.
Yeah.
Emily, how about you? Questions?
I don't think so.
Okay, pregnancy. What happens if the ovum is fertilized? So the sperm contacts the egg. It's very clear that the egg and the female reproductive system lay down peptides that actually guide the sperm to the egg. So the sperm, when it enters, there's two horns of the fallopian tube. The majority of sperm go up the fallopian tube with the egg, and it's clear the sperm also have in its membrane olfactory sensors. And if you use secretions of the uterus and fallopian tube from a pregnant female on sperm, the sperm will follow it around a petri dish. It's really quite amazing. So the sperm actually sniff out the egg, actually.
That's cool.
Yeah, it's remarkable. So the majority of the sperm go up, they know where to go, and they don't actually get lost. They know where to go. The video that you watched made it seem like the sperm just don't know where to go. They actually do. If they don't, it's because they're defective. And it's clear that a percentage of the sperm are defective, particularly under certain conditions. I don't know if you've discussed this in any other classes, but we talked about anabolic steroids decreasing fertility, but illicit drugs will too. Opioids will. Marijuana. Marijuana can actually drop your fertility substantially. Reasons for that are not completely clear, but if you do enough marijuana, it can really drop your fertility. So can cocaine. So can crystal meth. Those are all things that'll drop your fertility. Heavy metals, of course, heat. There was a study that looked at workers who worked in, there's those housings where it's like a greenhouse thing you work in for stuff that has to have sunlight, but you go in and you have to work on it. And you go in, you sweat a whole lot. You're under high temperature. What are those places called? It's like a greenhouse, but it's greenhouses are ventilated, but these aren't very well ventilated. It's to make certain things. Solar panels, I can't forget, but these workers are in high temperatures all day, and they have to take hydration breaks every 15 minutes or something. Those workers, if you measure their sperm counts, their functional sperm counts are way down because of the heat. So heat will do it too. So we're sitting in a hot tub and blah, blah, blah. In fact, fever will reduce sperm counts. So will actually a really, really intense workout. That can do it too.
So
So
that can be like short term.
yeah, it's short term. It's short term. Just heat is inimical to the development of mature sperm.
So have there been like any studies done on like whether there's like fewer pregnancies in summer then, or like anything like that, or like people who live in really hot states or anything?
Yes, actually. In hot states, fertility goes up in the colder months.
Interesting.
Yeah.
Which is counterintuitive because it's clear from other research that I don't read nearly as much, but social science research shows that couples have more intercourse during the summer months, but there are more conceptions during the winter months, which tends to confirm the heat theory that I'm propounding here.
That's really interesting.
Yeah, it's interesting. And I found I was directed to those papers by a social work major.
Makes sense.
Those aren't papers I read regularly. Can you imagine doing interviews like that?
Yeah, that'd be weird.
No
Yeah.
thanks. No
Yeah.
thanks.
I really want to talk to people about that.
Yeah,
No.
give me a lab bench any day of the week.
Yeah.
Yeah, I'd rather talk to my test tubes than that. Yeah, so let's see. Oh, yeah, pregnancy. So the sperm contacts the egg, you get the acrosomal response. That's in the slides, read about that. You get fusion of the sperm with the egg. The egg then undergoes very profound metabolic changes. The egg also releases a series of nearby granules that modify the cell membrane and prevent polyspermy, prevent fertilization with more than one sperm. And then the egg, which is quiescent metabolically, gets kicked into high gear. It completes meiosis 2. It sheds the second polar body. It modifies the sperm pronucleus. It modifies the egg pronucleus. The two pronuclei come together and they undergo the first mitotic division. And these are called the cleavage stages. First cleavage is 22 to 28 or so hours after the beginning of fertilization. Like we emphasize, fertilization is a process. It's not a single step. That process of the fusion, well, the touching of the egg and the sperm, then the fusion, and then the joining of the genetic material, that whole process is fertilization. It's a process. And so life begins at conception. You have to really ask, what do you mean by conception? And when someone says fertilization, you have to also say, what do you mean by fertilization? You mean because sometimes fertilization goes wrong and you don't get a embryo, you get a monster. So my belief is that human life begins at the end of fertilization. Conception is the end of fertilization, that process of fertilization. That's how I would define conception. That's for me when human life begins. When this human person is formed, when it now has a right not to be harmed, so on and so forth. So if you're interested in reading more about this, I wrote a book about this called The Stem Cell Epistles. There's a whole chapter on there. Plug my book while I'm at it. Anyway, so the egg, and if the egg isn't just staying in place, it's moving down the fallopian tube. So it divides, first cleavage, second cleavage. The second cleavage is not synchronous. You get 3-cell, 4-cell embryos. Then there's a 6, 7, 8 cell stage embryo. Then about 12 cells, the blastomeres undergo compaction, where they now start to adhere to each other very closely. Compaction creates two populations, cells on the outside, cells on the inside. The cells on the inside stay round, have a high nuclear to cytoplasmic ratio. Cells on the outside flatten, have a low nuclear to cytoplasm ratio. Cells on the outside become the trophoblast cells. They'll make the placenta. Cells on the inside become the cells of the inner cell mass. They'll form the embryo proper. The cells on the outside pump salts to the interior of the embryo. Water follows, the embryo will cavitate. Now, about 5 days after the beginning of fertilization, you have a blastocyst with trophoblast cells on the outside, intercell mast cells on the inside. It'll reach the uterus about late day 4, early day 5, and it's competent to implant into the endometrium day 6. So between late day 6, day 12, the embryo can implant. If it doesn't implant within that window, it'll never implant and it will die. That's why making an artificial uterus is quite difficult, because implantation is a process that began very early. To get it to unimplant and then reattach after this very complex process of implantation and placenta making that required both the embryo and the endometrium, that's a very hard process to redo. So that's why making an artificial uterus is such a hard thing to do. All right, so stop, questions? No? All right, implantation is really complicated. It's beyond the scope of this class. Suffice it to say, there's immense crosstalk between the embryo and the endometrium. The embryo signals, the endometrium signals back. Then the embryo signals again, the endometrium signals back. The embryo scratches the endometrium's back, the endometrium scratches the embryo's back repeatedly. And so it's like the ultimate political arrangement, so to speak. So the embryo attaches, the endometrial cells will undergo the decidual response. They actually grow around the embryo and embed it inside the endometrium. And then the trophoblast cells become two populations, one that fuse into a multinucleate population called the syncytia trophoblast, the others that become cellular, stay cellular, and become the cytotrophoblast cells. The syncytia trophoblast cells will invade and digest tissue, providing nutrients for the embryo. They're attracted to oxygen, so they'll be attracted to blood vessels. They'll erode the blood vessel, make a little pool of blood. These are called blood islands. And these will be the first areas where you get gas exchange between the embryo and the endometrium. And then the cytotrophoblast cells will move into that area. They'll make the first blood vessels, and those are the beginnings of the umbilical veins and arteries. The embryo at day 14 will undergo this process called gastrulation. It now goes from being a single layered structure to a 3-layered structure. The ectoderm, mesoderm, and endoderm. The endoderm forms the GI tract and associated structures. Mesoderm forms skeleton, muscle, glands, blood vessels, heart, stuff like that. The skeleton, the ectoderm will form the skin and then will undergo neurulation and form the nervous system. That starts at day 14. Embryonic development goes from the beginning to the end of the 8th week of development, day 56. Books will talk about the pre-embryonic stage. The pre-embryonic stage does not exist. The pre-embryonic stage was made up as a political stunt to justify embryonic cell research. There is no pre-embryonic stage. The whole thing is embryonic development. During the embryonic development, all organ systems are formed. They're not mature, but they're all formed. So at the end of 8 weeks, you have all your organ systems made. The fetal stage is maturation of those organ systems. And the podcast I did yesterday focused on the physiological changes that occur in the woman's body during pregnancy, and what have you. So take a listen to those. If you have friends who've been pregnant, they've probably shared with you some of their woes, so to speak. Some of that is just changes that accommodate the fetus as the fetus grows. Other changes are due to estrogen and progesterone levels. And blah, blah, blah. If you have questions about those, you can ask me. I'll let you listen to those podcasts first. Let's see. Okay, so delivery pretty clearly occurs because the fetal adrenal gland starts to make cortisol, increasing levels of cortisol. That cortisol now is converted by the placenta into estrogen. And the estrogen of pregnancy is not estradiol, it's estriol. Estriol. The estrogen will cause the myometrium to make oxytocin receptors and will also cause the expression of gap junctions in the myometrium. So now the myometrium gets more connected and it gets 100 times more responsive to oxytocin. Progesterone will prevent the myometrium from contracting. However, with increased estriol, because of the production of cortisol from the fetal adrenal gland, now you'll start to get little, do you know what Braxton Hicks contractions are?
I've heard of them, but I couldn't tell.
They're the so-called practice contractions. So women who've had a baby already usually won't even notice them. But a woman who's not delivered before, she'll start to notice them. They may be as much as 2 weeks before delivery. My wife started to notice hers just the night before. And they're kind of more of a nuisance than anything else. They don't hurt. But that's due to that increased estrogen. And then the practice contractions will start to increase in intensity. And 2 things happen. The cervix will start to efface. That means the cervix is going to go from a neck-like structure and it'll start to flatten. That's what effacement means. It flattens. As it flattens, the baby now starts to push on that cervix. You have mechanoreceptors in the cervix. When you activate those mechanoreceptors, that activates the hypothalamus to make and release more oxytocin. That increases contractions. You have a classic positive feedback loop. And so the first stage of pregnancy is a so-called latent phase, where you're in that Braxton Hicks gradual effacement phase of the pregnancy. And usually the OB or the delivery nurse will do a pelvic exam. They'll try to insert their fingers through the cervix, and how far they can insert their fingers will be determined. They'll say the baby's at -1, -2, -3. Minus has to do with how far you can insert your fingers. If they can't insert their fingers at all, then the baby's at 0. And if the baby's head is starting to protrude, then it's a +1, +2, +3, +4, so on and so forth. That's what those terms mean. And labor and delivery nurses are very skilled at that sort of thing. I always wondered, that has to be a judgment call. They said, no. They said, you know. You've done enough times, you know. And I was like, well, how do you, is it this far? Is it, knuckle or something like that? They said, no, you just know. You know. So I said, okay, I'll trust you. That's what those terms mean. But eventually, the Braxton Hicks contractions, you'll have about 3 to 5 minutes between contractions. As you get toward the end of the latent phase, now your contractions get much closer together. But the big kicker is they get more intense. And they get to what OBs, they call them good contractions, although a mother in labor won't say there's anything good about them because they hurt. They're actually called the powerful contractions of labor. And now you're in the so-called pushing phase of labor. And it's the 3 Ps: pushing, propulsion, and position. And the baby, it depends on the baby's attitude. And that's not, whether the baby's pissed off or not, that's how the baby's positioned in the womb. So baby needs to be head down, and also needs to be turned so that the baby's spine is against the mom's spine. If the baby's spine is turned, that makes for a harder delivery. If the baby's head up, that's breech. Now you have to try the turn to baby. Midwives, labor delivery nurses are very skilled at doing that. If the mother's uterus is small, it gets harder to do. Also, the risk of uterine rupture goes up. So it's a judgment call. There's a woman in my Bible study who's very anti-C-section. She thinks if a doctor ever does a C-section, she's committed a mortal sin for which he or she will never be forgiven. Sometimes C-sections are just necessary. It's just, you're not going to get the baby turned. It's not going to happen. So C-sections just have to happen. So the baby's head down, and then the chin needs to be against the chest for passage. That's the other feature. And then once the baby's head gets through and the pubic symphysis has been softened because of progesterone, and the ligaments that hold the cardinal ligament and all those that you guys know about those, they soften. So now that pubic symphysis now will open. In fact, that accounts for a lot of the pain while you're pushing. The baby's head goes through, and then usually one shoulder will poke through, and then the baby will usually have to rotate to get that other shoulder through. And so the baby comes through, there's usually a rotation, and then a rotation to get the other shoulder through. Once that second shoulder pops through, it's usually out it comes. And then you have the placental delivery stage. That sometimes is part of the recovery stage, or it's a separate stage, because there's usually, the mom usually doesn't have to push anymore. And sometimes, usually, the mom is given an injection of Pitocin in the umbilical vein because you have increased contractions that will cause a contraction of the myometrium so that the placenta is delivered. And then the placenta is put in a dish, and a human placenta is shaped kind of shaped like this. It's got a wide base and a tapered head. And if a woman is past term, parts of it are calcified usually. You have to make sure you get the whole thing. If you left a piece of it in there, that gets necrotic and gangrenous. And that can be very dangerous. So you have to make sure you get the whole thing. You put it in the dish and you make sure that it's the right shape. And that's something that, again, OBs are very good at. Midwives are very good at. And then the estrogen progesterone levels now drop like a rock. And something estrogen and progesterone did that we didn't talk about is they influence breast development. So the breast, your book has a really good picture of the lobules of the breast. You have these alveoli that are lined with milk-secreting epithelia. They're drained by a milk duct that then is drained by a lactiferous duct that goes into the nipple. That tissue, under the influence of estrogen and progesterone and prolactin, now becomes very much differentiated. You have increased fat deposition. The alveoli now swell up, they bud, they get more of them. And prolactin will cause milk protein and milk production, but lactation is inhibited by high levels of estrogen and progesterone. After estrogen and progesterone levels drop because of the delivery of the placenta, now the breast fills with milk. And then when the baby suckles, you again have a positive feedback loop. The mechanoreceptors on the nipple send a signal to the hypothalamus. You get more oxytocin. The lactiferous duct contains myoepithelial cells that contract in response to oxytocin. That's called the milk letdown reflex. And so you get milk pumped by the contraction and by the suckling of the baby. As the baby ages and the mom breastfeeds more, actually just hearing the baby cry or seeing her baby will actually cause lactation to start. And especially if you've had your second or third baby, sometimes just you wake up hearing the baby cry, you start to lactate, which moms have told me can be very irritating. But that's just, you get cued to your baby, and it's more than just suckling, it's visual and auditory signs as well. My wife, our first baby, she'd just go, something you do as a young mother is you take naps. So when the baby naps, you nap. And if your kid's not a good napper, like my first kid, you almost get no sleep. I thought Rachel was going to be the death of us. Next two, wonderful sleepers. Rachel would do this. She'd go, fight
no.
sleep. She was a legendary sleep fighter. It was crazy. I've never seen a kid do that.
That's funny.
God, kid, go to sleep. You're tired. It just, yeah. Anyway, so she'd watch Rachel sleeping, and then the second Rachel woke up and looked at her, she started lactating. So, and that can be very individual too. So that's due to the interplay of prolactin and oxytocin. Prolactin is inhibited by constant secretion of dopamine by the hypothalamus. But the high levels of estrogen and progesterone prevent the secretion of dopamine. And then the milk letdown effect, that prevents dopamine production as well. Now, when you wean your child, you stop nursing. Now that reflex stops. That's why when you stop nursing, to start nursing again can take a while. Also, during wartime and during times of stress, you can stop producing milk as well, because now that stress will start the production of dopamine again. Did you guys watch the Netflix series Waco?
No.
During you guys weren't alive then. You guys were born what? 2001,
99.
99. Yeah, that was during the Clinton years. You guys weren't alive during the Waco incident. It was a bad mishandling of what should have been just a simple arrest and search of a compound in Waco, Texas, by this whack job named David Koresh,
Oh
who
yeah,
thought he
that's like
was,
he had a cult, right?
yeah, that was seriously mishandled by the ATF and then the FBI after them. Janet Reno should have lost her job, but she didn't. It just, when you cock something up that badly, anyway, the women in the compound who had, many of them were mothers who were nursing their babies, all of a sudden their milk dried up because of the stress. And there's a scene where the moms have their babies and they're talking to each other and they're saying, my baby's hungry. Do you have any milk? Because my milk dried up. And the moms are all shaking their heads. And that's stress cessation of lactation, it's called. Stress-induced cessation of lactation. And that's due to stress hormones, usually glucocorticoids, or activation of the sympathetic nervous system. That will decrease your lactation. Also, athletes who go back to hard workouts sometimes have to time. They usually have to pump, and then they have to bottle feed, because after a hard workout, they may have trouble making milk. Although that's very individual. You know who might know more about that is Beth. If Beth is in the lab there, she might know more about that, because I know Beth was a runner in college, and she continued to run after she had kids. And she'd know more about that than I would. But I know sometimes that's very individual, but that does happen. So Beth, you there?
Oh, here, I have my head.
Hey Beth,
Yeah.
what do you know about women athletes who go back to hard workouts and then try to breastfeed? I know sometimes they'll stop making milk, but I've also read that's very individual.
Yeah, I don't know, but I would have to say that I could see that happening, but maybe not to everybody, like you said. So
Because when you had your kids, you were still running, right?
I was not running. I was working out, and I didn't have any issues.
Okay,
So,
okay.
yeah. I wonder if it would be more if women already maybe struggled producing milk in the first place, and then.
That's possible. That's
Yeah.
possible. Probably also depends on how hard your workouts are.
Right.
Because after you've had a baby, you definitely have to ramp up. You
Yep.
can't just go back to what you have to, because your ligaments and stuff are still loose. You have to
Right.
tighten them up first, and it's probably very individual.
Yeah, and I had a C-section, so I had to recover from that. And so mine
okay,
was definitely a very gradual return.
okay, okay,
Yeah.
okay.
Yeah, that's an interesting question though. So
That has been worked on. That has been researched.
yeah, I'm going to look at that because I definitely counsel some women postpartum. So I'm going to look into that some more.
Yeah, because postpartum exercise is huge with avoiding postpartum depression.
Right. Yeah.
And so there's been lots of exercise programs, but that was a complication, was exercise-induced cessation of lactation, at least in some women,
Okay.
because depression will dry you up too.
Oh, yeah. So, yeah.
Yeah,
Difference. Yeah, the stress
Yeah.
on the body.
okay.
Yep.
Well, okay, that was a nice aside there. So, yeah, well, you guys are both interested in healthcare. You may have an interest in that someday. You'll probably counsel women postpartum. It's an important part of healthcare because that group, I think, is a neglected population in healthcare. They really are, as Beth will testify to. Okay. Anyway, let's see what else. I think I hit everything.
Okay.
Oh, yeah. Prolactin. Prolactin feedback inhibits the production of gonadotropins.
Okay.
That's why breastfeeding can be a contraceptive to some extent. It's not perfect, but that's why women who breastfeed will tend not to conceive. That's also why noble women tended to have wet nurses, because noble women wanted to stay pregnant. They wanted to deliver lots of kids. That's why they had wet nurses.
Okay.
Little piece of historical thing for you. Emily, any questions or any of that? Or
I had one just kind of random about C-sections. So is there a point to where, like, say the baby is breech and they should do a C-section, is there a point to where it's too late to do one or they can't do one for some reason?
Yeah. You're starting to get outside my area of expertise. From what I've read, which is, I've got these couple of books here. It depends on the level of fetal distress.
Okay.
Once the mother goes into labor, the labor will tend to cause detachment of the placenta. Also, if labor causes the water to break, the baby is fine for a few hours, but if the baby is in the uterus and the placenta detaches, the baby will start to go into distress, fetal distress, the heart rate will start to drop, and then you need to get the baby out, usually via C-section. It tends to vary. So some OBs are very conservative. If the baby is breech and you try 2 attempts to turn the baby and the baby just won't turn, then it's like, okay, C-section.
Yeah.
That's just it. Others are less conservative. They're no, we can do this. C-section is last resort.
Yeah.
The issue is that the estrogen and progesterone tend to cause the muscles of the abdominis rectus to separate.
Yeah.
And because they separate, long periods of labor increase the risk of tearing. And if you get tears, that's going to make future pregnancies really difficult.
Yeah.
So, also, if a mother has already had a C-section, the risk for uterine rupture goes up.
Okay.
And if the uterus ruptures, the mom's risk of hemorrhaging go way up.
Yeah.
And uterine hemorrhage, you've got minutes to be able to save the baby and the mother.
Yeah.
So that's a medical emergency. And again, this lady in my Bible study, she thinks a lot of this is driven by insurance companies and lawsuits and things like that. She's partially right, but the research is clear. The risk of uterine rupture goes up after a C-section. So a VBAC, vaginal birth after a cesarean section, VBACs have increase of uterine rupture. They do. That's a fact. And so yes, your insurance rates go up in that case, but you have to take into account the risk to the mother. And yes, there are midwives and OBs who do VBACs after C-sections perfectly well. There's no problem, but you're weighing risks usually.
So will
So
some people, sorry,
go ahead.
will some people, if they already had a C-section and they're going to deliver their like second baby after a C-section, will they just decide like before that we're just gonna do
Yeah, if the mother says, that first time, I'm going to go through that again, or I'm just going to have a C-section again, probably.
Yeah.
Especially if the baby hasn't turned, then the doctor will usually say, yeah,
We'll
that's
just-
fine. We'll just give you a C-section and you'll be all right. Some professional women will actually schedule C-sections so that they can have the baby and then go back to professional life. Others, there's usually the hospital will have an algorithm for when you do a second C-section, and you have to respect that because it's weighed by medical risk to the mother, but also the insurance is part of that too. It really is. So it does factor in, but the medical risks do dictate insurance risks. So they're not completely detached. So let's give doctors some benefit of the doubt. They're not just completely basing this on lawsuit risk and what have you. So, yeah, so it's fatal risk and then risk to the mother. Also, if the mother has a so-called, some women have a short cervix, they have to be on bed rest. In cases like that, giving birth might be really difficult. And the doctor might just say, it's time for C-section. Also, if the mother's over 40, or if the mother is 40, they may just say, you need a C-section. Labor is going to take too much out of you. We need to give you a C-section. My wife had our oldest at 40, and she was fine. So that's very individual as well.
Yeah.
Also, women are having babies later in life, so you're seeing more C-sections. The US gives more C-sections than Europe does, largely because the OB profession here is more conservative. we
Mhm.
also have less maternal death. So
Yeah.
when the anti-C-section people quote that statistic, also tell them, we have less maternal death and our mothers are older here.
Yeah.
So how
That
about you
says
quote that statistic too?
Yeah.
Okay, any more questions?
I don't think so.
I think I'm good.
All right, Emily, I don't think you're on. I told Kayla I'm not going to put the test on Blackboard probably because the formatting, it doesn't accept the formatting. And so I
Okay.
literally have to type every single question instead of just pasting it. So
Oh,
it's going to take me hours
yeah.
to do that. So I'll probably just stick with a paper test. You'll get the paper test by email. You'll take the paper test and then re-email it to me. And that you'll just take the test on camera and that's the way we'll do it.
So what time is it?
Test is Friday. I think it's at 1,
So we'll all, like the whole
That was
class will get on a
10:30 to
Zoom?
12:30.
12:30, 12:30, okay, 12:30, yeah.
12:30 on Friday?
12:30 on Friday.
Well, I thought the exam schedule said it was 10:30 to 12:30.
Oh, okay. 10:30, 12:30
Okay,
Friday.
so will you send a Zoom link then?
Yeah.
Okay, and then we'll all just get on and we'll have our test already printed off.
Yeah, or you could, so you could do it, scan it, or just take it on the computer and then
Okay.
send it back essentially.
Okay,
It'd be either case.
okay.
Is it going to be the same as the past test? Like,
Oh, yeah.
I don't remember how many
And
questions.
same
all multiple
format as a
choice.
practice test.
Okay.
Did you get the practice test?
Yes.
Yep.
Same format as that.
Okay.
Sounds great.
Okay.
Yep, thank you.
Okay,
thank you.
Emily, I'm gonna head to the gym after this, so I'll probably see you.
Yep,
So,
I'm here.
okay, well, Kayla, you're there too, so I'll probably see you.
Yep.
Yeah, I am.
Okay,
All
folks.
right.
See ya.
Thank you. See ya.
Yeah, bye.